Let's welcome our next session, The Aging and Brain Challenges. Are we at the cusp of breaking through? Featuring Howard Fedorov, the Chief Executive Officer at Aspen Neuroscience, Howard Phillott, Founding Executive Director and Chief Scientific Officer for Alzheimer's Drug Discovery Foundation, Irene Griswold-Prenner, Chief Executive Officer and Chief Scientific Officer at Nitron Bioscience, Rudolph Tanzi, the Joseph P. and Rose F. Kennedy Professor of Neurology at Harvard Medical School, Vice Chair of Neurology at Mass General Hospital. The session leader here will be Barbara Dalton, Managing General Partner at Pfizer Ventures, Vice President of Worldwide Business Development, Pfizer.
All right, then let's jump into it. The topic of the session is the aging and brain challenges. Are we on the cusp of breaking through? Thank you to all the panel members for joining me this afternoon. I just wanted to make a comment about the topic we're discussing so that we can focus in on what I call the diseases of the developed brain. I think that for the most part, neurodegeneration is in contrast to a lot of the diseases of the developing brain, so the psychoses related diseases. And today's discussion is going to focus on the diseases of what I'll call the developed brain. And I'd like to start that discussion by asking Rudy to sort of give us a little bit of a framework to sort of move into talking more specifically about the biggie of neurodegeneration, and that's Alzheimer's disease.
Yeah, it's interesting to think about developing brain and non-developing. If you look at the genetics of Alzheimer's disease, the first genes brought us to amyloid, and then we learned that the amyloid comes very early, and we have to treat that probably pre-symptomatically to do it the best way. And then the newest genes from GWAS studies have been common variants that are involved with neuroinflammation. But I just want to mention that we're seeing an increasing amount of rare variants in genes that bring us back to neurodevelopment and synapse maintenance. So I think we're going to see a mixed bag when the genetics is all done between genes that affect the pathology itself early in life, genes that affect neuroinflammation once the disease really takes hold, and a growing number of genes that affect synaptic integrity and neurodevelopment. And then we have to then do the right thing in terms of treating the right pathology at the right time, in many cases pre-symptomatically. I think this will be a big theme going forward for this disease.
So pre-symptomatic treatment versus post-symptomatic treatment. Interesting. So what are some of those factors? Howard Phillott, I'm going to throw this your way because I know that ADDF was instrumental in some of the very early studies in trying to identify factors that appear early or conditions of which that can be associated with propensity to develop disease or protect from disease. So would you like to comment on that in terms of some of the early things we can see before we get into major disease and neurodegeneration?
Yeah, I kind of would like to add that I don't see it just as the developing and the developed brain, because to me, the developed brain implies a normal brain. And I think what we really need to think of strategically from the point of view of even drug development is translating the biology of aging into new drugs, because Alzheimer's is the leading risk factor is aging. And we know so much about aging biology today. And so much of that biology has never been translated into effective pharmacology, even in terms of new emerging strategies that are pleiotropic and sort of comprehensively attack the effects of aging on the brain, such as senolytics. And when we talk about treating the pathology, I think that's a very narrow approach to drug development, because we know that the senile plaques, as Alzheimer's originally called them, is composed not only of beta amyloid protein, but many other misfolded proteins that we've just gotten the tip of the iceberg, like TDP43, for example, and as an emerging pathology in the aged brain, very common in 90-year-olds. Obviously tau, and I think the real breakthroughs are coming right now in terms of our biomarkers, which are really going to enable us to translate the biology of aging into new therapeutics. So we have amazingly a blood test for beta amyloid, and we're going to have a blood test for phospho-tau, and we have all this amazing neuroimaging. So we've learned a lot about the aging brain, and I think that's really key to to how we go forward. And I think genetics, as Rudy would say, it's probably going to be a polygenomic effect that leads people to greater risk. And in some cases, maybe not, like TREM R47H, where you have a single gene that gets, impairs the inflammatory response. So it's just so much interesting things. I won't go on. I don't want to take up too much time.
Is there a way to identify the patients that should be tested early? Should I have been tested when I was 40? How would you choose? Is it a genetic choice? Because we can't test everyone. Let's put it that way.
Well, I think the goal from a public health perspective, if you think about how do we underwrite healthcare? Let's just take Medicare and what's recommended for patients when they come in for an annual visit. It's likely that part of that evaluation, one could have a very low-cost test that's non-invasive that will actually begin to delineate and maybe build in time a trajectory of risk. I think that's one goal. But that starts later in life. With regard to a 30 or a 40-year-old, I think that it's very hard to justify today actually extending that much earlier in life, although I'm sure that once the newer science emerges that suggests that you might be able to detect, even among those much earlier than a 65 or a senior, the propensity then to go on in years hence to develop Alzheimer's, or for that matter, PD. I think you have to think about, on a population basis, does this make sense? And my belief is currently that it's very hard to justify on an economic basis, and also there will be not 100% sensitivity and specificity, so you'll have false positives and false negatives. And I think the idea of having personal trajectories where you have multiple data points may give us greater confidence, particularly when you serially measure the same thing and you know it's going in a direction that's suggesting that risk is increasing.
So Barbara, if I could comment on that. I think this revolution in digital technologies is going to address some of Howard's concerns, which I totally agree with, in terms of when and how is prevention cost effective, but also how we get multiple data points through either passive or active monitoring. And Howard mentioned sleep. There are ways now, that technologies that we've seen at the ADDF through our diagnostics accelerator and our digital efforts, where people can look at sleep. I mean, we already know this on your Apple Watch, and all these very simple technologies that can measure even REM sleep with very simple technologies through active monitoring or passive monitoring. Even more sensitive, I think, is speech analysis, analysis of computer use, language analysis, movement analysis, all these other methods that can be used passively. And so it makes screening a lot easier, or people don't actually have to enroll if they just participate in passive monitoring. So I think part of the way forward in terms of early detection, particularly for prevention, but also as outcomes in clinical trials, we'll use these really advanced, amazing digital technologies that are out there, even multimodal technologies that incorporate 4 or 5 of the things that I mentioned, including, of course, the simple ones like cognition and function, which can also be measured. So right now in clinical trials, people come in once a month or once every 3 months for cognitive testing. Now we can do, cognitive testing on a passive basis on somebody's iPhone, once a day, and get so many data points. So I think I agree with Howard. There have been recent articles on looking at the cost effectiveness of prevention. And most of the time, prevention actually isn't cost effective. It's something desirable, but it's not necessarily having a public health benefit when there isn't a low-hanging fruit. And I think that's a very important point.
Rudy, I'd like to take this to you and ask you about the use of early diagnostics as far as our clinical development of therapeutics is concerned, because outcome studies just cannot be afforded anymore. I mean, it's one of the reasons why a lot of companies have decided to leave the field in terms of identifying drugs until it becomes easier to identify patient populations and to determine whether you have a drug which is going to do something. So what would you say to the regulatory aspect of using these early markers?
Yeah, I mean, right now, the elephant in the room is that with neurodegenerative diseases like Alzheimer's, we're reactive. We wait till the brain degenerates to the point of dysfunction and then try to bring it back. We don't do that with diabetes or heart disease or other age-related disorders. And part of the reason is that every year we don't have a checkup from the neck up for the brain. So getting back to Howard and Howard's comments, I see a staged type of process where we will figure out how to assess brain health on an annual basis, digital technologies, maybe a general test for neurodegeneration like neurofilament light chain. And if that looks off, then you go to an A-beta test and a tau test. And if that looks off, maybe you might want to do a PET scan just to dig in a little further. Whatever the insurance company requires to say now it's actionable. And then you need to have the drugs that hit the early pathologies. And I think what's emerging is that proteinopathies, amyloid, tangles, Lewy bodies, TDP-43 come early. And they trigger enough neuronal cell death over time to trigger neuroinflammation. But many studies show neuroinflammation is killing 10 times more neurons than the original proteinopathy. And if you want to help a patient who's suffering now reactively, you have to hit neuroinflammation. But the future for my 12-year-old daughter will be when she turns 30 or 40, she'll get these tests done. And if necessary, serial tests done to see what's going on. And then we're going to do secondary prevention to hit these proteinopathies like amyloid and tangles and Lewy bodies early on before the brain degenerates with neuroinflammation. So how do we get there? If the FDA requires us to show that a drug that stops amyloid or tangles or Lewy bodies makes a patient better in memory or movement, we're not going to get there. Imagine if in order to get statins, we had to show that somebody with congestive heart failure who just had an MI takes Lipitor and gets better. I mean, you do take a cholesterol drug, right? If you're sick, but you don't necessarily, but it's not going to necessarily make the heart better if you already have congestive heart failure. And Alzheimer's disease, you may take an amyloid drug or a tangle drug when you already have the disease, but you need to hit neuroinflammation. So how do we take care of my daughter when she's 40 years old and somebody says, it looks like you have more amyloid or tangles in your brain. I hope that's not the case, versus the population for your age. Well, you're going to have a drug that hits that proteinopathy. So I think the good news is the FDA in February 2018 had draft guidelines. They said if we can all as a field agree on biomarkers that indicate pre-symptomatic Alzheimer's has begun, then they will allow trials that move those biomarkers. And if we have drugs that safely move those biomarkers, be it A-beta or tau or NFL, neurofilament light chain, that they would consider approving them and then depend on real-world evidence to see if it's moving the dial on incidence and prevalence. I think this is the only way forward, because otherwise we're only going to have drugs that are reactive and not the drugs we really need for the future that are proactive, that are actionable steps after you find through early detection that you're getting into some trouble. And I would only add that the role of genetics is to predict early who needs to have detection earlier than usual. So let's say detection is usually going to be in the future at 40 or 50. If you have a family history that's earlier, you might want to get that early detection at 20 or 30. So early prediction through genetics, early detection through biomarkers, actigraphy, like Howard was mentioning, digital technologies and imaging, but then actionable steps where you actually prevent those early initiating pathologies, mainly the proteinopathies like amyloid tangles and Lewy bodies. But the FDA has to allow us to get to that point. And that means trials that move biomarkers we agree on, and then trusting that these will help in preventing the disease with real world evidence over time.
I want to come back to the neuroinflammation with you, Howard Phillott, and you, Rudy, because I think there's a chicken and egg question here. Which came first? Inflammation, proteins, inflammatory response to the proteins? So isn't it a chicken and egg where the role for neuroinflammation comes in? Because we know inflammation in general increases with age, probably due to some dysregulation of the immune system, et cetera. Things aren't working as well as they did before. Or is it, did it start with that and prevent cells from doing the things they're supposed to do and cleaning up the proteins that are accumulating in the brain?
Yeah, I mean, I think you're making some good points, and it is another part of the puzzle, but sort of a different way. The way I think about it a little differently than the way Rudy has, but I think Rudy's right. But as you mentioned, Barbara, inflammation is a key feature of aging, and it's systemic inflammation. It's from all the joint degeneration, the vascular injury, the multiple diseases that we suffer with aging. We have systemic inflammation, and we know that systemic inflammation does get into the brain through cytokines and triggers and primes microglia. So systemic inflammation can also cause not only neuroinflammation by priming microglia, but also there's been a number of studies, some of which we did in my lab like 20 years ago, showing that cytokines in cell culture increase the amount of beta amyloid that cells produce. So there's also an effect of systemic inflammation on increasing the number of misfolded proteins that are produced. And part of that goes to another issue that I think is going to be the next generation of therapeutics, because if we try to pick off one or two of these misfolded proteins that are characteristics of the pathology, one or two of these at a time with an antibody, we might end up with, requiring, as we identify more and more of these misfolded proteins in the plaque, somebody's going to need a cocktail of 10 different antibodies, whereas the real problem with aging is a failure of autophagy. And that's really the pleiotropic approach to drug development, I think, for all these diseases of misfolded proteins with aging. The problem has been that autophagy has been a very difficult biological process to target for drug development. But I think we're starting to get there in terms of very specific stages of autophagy that can be druggable and can be, a more pleiotropic approach to the misfolded proteins that occur.
So I'd like to move to the aging process, normal aging, pathologic aging, whatever the case may be. But in the aging process, we do have a change in our anti-inflammatory, responses too. And that leads to accumulation of the proteins that we've been talking about, but also some modified proteins. So Irene, would you like to comment on your favorite area of emphasis in terms of the way those proteins are modified and the impact upon senescence, which is a topic I'd like to come back to because Howard mentioned the senolytics, but I'd like to move to that specific aspect of inflammation in terms of the work that you're doing.
Sure, I'd be glad to. I think that there's several components that contribute to both the aging and the neurodegenerative process. We've heard about several of them, the impairment of autophagy and senescence and even telomerase length, et cetera. But one of the things that we've been studying at NITROM, or the thing we're studying at NITROM, is the oxidative stress pathway. And up until this point in time, it's been proposed that oxidative stress, and particularly an event called nitration, which is sort of the major endpoint of a lot of oxidative and nitrative stress, is a chemical reaction. If it's a chemical reaction, there's really not much you can do with it. Spin traps have been tried and high levels of vitamins, et cetera, but there's such high concentrations of oxidative stress in our aging brain that they have been ineffective at this point in time. And what we realized early on is that there was a hidden class of enzymes that are involved in the oxidative pathway, and that's enzymes that catalyze nitration. And it's been shown by a number of groups that if you nitrate alpha-synuclein, you will drive its aggregation and you drive death of the neurons that are affected in Parkinson's disease. So our goal at NITROM was to be able to determine whether we could see if we could identify the enzyme that catalyzes nitration of alpha-synuclein. We were able to do that, and we're able to show that inhibiting it or knocking it out drives the type of neurogeneration, or inhibits the drive of neurogeneration that is relevant in Parkinson's disease. And in the process of identifying this enzyme, which we call synuclein nitrase, we found that there's actually a class of enzymes that catalyze events across a number of not only aging-dependent diseases of neurogeneration, but also any aging-dependent diseases. So we found everything from the neurodegenerative diseases, including ALS, Alzheimer's, Parkinson's, into even certain types of aging-dependent oncology, fibrosis, et cetera. And that's the part that we're particularly excited about in terms of working with a class of enzymes that no one has known about before and being able to drug them. And I think it'll be a complementary piece to what is already being done in the field, the anti-A-betas, anti-synucleins, anti-taus, the autophagy inhibitors, neuroinflammation. I think when you bring all these together, I think that's where we're really going to have the real advancements in being able to treat these diseases that have been going on for decades.
Well, we've talked about how we'd all like to treat earlier, and there's a lot of approaches to try to go earlier, but what about later? And I'll go to you, Howard, for this question. Are we going to be able to replace any of my neurons that I'm losing as I age or as I develop any neurodegenerative diseases? Are there approaches which are really, truly going to be earth-shatteringly, that will be able to change the decline, not only just change the slope of the decline, but potentially bring back activity?
I think there's ample reason to be optimistic. Start with Parkinson's disease. Most people think that Parkinson's disease is simply a movement disorder. It's clear that that's a prominent feature and probably early on brings most of these individuals to clinical attention. But we know a lot about the loss of the circuits. Rudy made the point, and I'd underscore it, that neurodegeneration is about loss of functional circuits. And in the case of PD, Parkinson's disease, at least the motor features are largely due to loss of dopaminergic innervation into a part of the brain, principally the putamen, but also the caudate. When those synapses are lost, it's because the cells where they reside in the midbrain, the substantia nigra, have died. At diagnosis, 50 to 60% of the cells are dead. So what we do know, and there's been fetal transplant studies, and now there's been 5 patients worldwide that have been treated with stem cell-derived dopamine neurons, that you can restore function. It's very early days, but it suggests at the very least that paradigmatically, you can restore the circuits that actually are lost in PD, and you may even buy patients time. And so part of the work that we do in the company that I'm part of is really doing this on an autologous basis, where you start with your own cell and then you transplant your own derived dopamine neurons. Alzheimer's is much more complicated. And I think that the challenge there is that it's so vastly much more synaptic losses occurred. I don't believe that a simple transplantation paradigm is going to necessarily work for AD. I strongly endorse taking on neuroinflammation as early as it can be detected. And I also endorse Howard Phillips' point that systemic inflammation often antedates neuroinflammation. So there may be a way to begin to think about that. But lastly, I'll just leave you with this. I think we know enough about the CNS niches that contain the cells that are stem cells. And so we know that neurogenesis, while it is highly variable, it still does occur in the older adults. I think one thing that would be very intriguing as we get further down the road and understand how to replace cells, can we replace them and have drugs that augment neurogenesis in the context of being at risk or manifest? And can those circuits be rebuilt de novo?
In vivo neurogenesis.
I think it's, I think it's not going to be only about growth factors. It's going to be about small molecules that are going to tweak some of the pathways that lead from being pluripotent or committed to ultimately then being able to fully differentiate and restore circuit function. So I think it's a bright future. And I think that while we don't know enough today to be explicit about what drugs, we do know enough about the stem cell biology to make certain predictions about what might be druggable that could, in effect, endow those niches with the capacity to make more cells that can then ultimately be functional.
Can I comment on that, Barbara?
Yeah, sure.
We had I totally agree with Howard that neurogenesis has so much potential. We had a paper in Science a couple of years ago where we took Alzheimer's mice, and it was very interesting. You know, if you have them exercise, they get better. You give them a running wheel, it's about as good as any drug as we've tried in an Alzheimer's mouse. And interesting, if you check who's exercising and who doesn't, The ones who sit and watch the others exercise don't do as well. So like watching sports on TV rather than getting exercise. So what we found was that exercise induces neurogenesis, right? So we used drugs and genetic therapies to induce neurogenesis in the mice. And the fact is they didn't get better. And we were baffled because we also saw that neurogenesis was in the Alzheimer's mice early on was also delayed. So when we asked what else does exercise do, and this gets to the growth factors, Howard, the exercise was not just inducing neurogenesis, but was inducing BDNF, brain-derived neurotrophic factor, plus irisin is another protein was inducing. So we just went step by step and we said, let's induce neurogenesis and add a growth factor. And at the end of the day, if we induce neurogenesis with one drug or genetically, and induce brain-derived neurotrophic factor, BDNF, with another drug or genetically, that mimicked exercise. Then the mice, even though amyloid was still there, neuroinflammation was still there, the cytokines were still there, the pathology was still there. But because there was neurogenesis, and I look at the BDNF kind of like miracle grow for neurons, of maybe it's helping these new little stem cells to survive in the battle zone of an Alzheimer's brain. And then the mice basically got better. So, I think first we have to hit the pathologies directly. I do, amyloid entangles early, neuroinflammation early and late. I think you can hit neuroinflammation in a patient later, and whereas plaques entangles probably not. But I think hitting neurogenesis and inducing that despite the presence of pathology is another good way to help bring these neurons back naturally in the hippocampus, where adult hippocampal neurogenesis does occur. So we're testing now supplements to do this, because there are some supplements that induce neurogenesis and induce BDNF, and we're trying those combinations because they're safe, and the idea would be if we can find a couple of safe natural products to do it, we might try to do some trials on that as well.
In my simplistic view of what's going on in the brain with exercise, I always thought that increased blood flow is very helpful because it's flushing things through the system, let's put it that way. And it helps in the cleanup of the accumulation of products that you don't necessarily want to hang around there. So I want to bring this back to senescence, because a hot topic these days is senescence and understanding why these, and I love the term, the zombie cells, are created that just sort of hang out there, and anybody can understand what a zombie is. That's not a living cell, and it's not a dead cell. It's somebody in the middle in suspended animation that hasn't gone through the autophagy, it hasn't gone through apoptosis, it hasn't gone through the routes that normal cells take to undergo cell death, and it's sort of stuck there. But how are we going to be tackling disease, in addition to autophagy, what else in the senescence world are we going to be looking at that may have an effect on neurodegeneration in a variety of diseases? Because once again, a broad pathology or a broad stimulus like inflammation in terms of what's going on there.
So if I could answer a little bit, or the senescent-associated secretory phenotypes of cells have been found in Alzheimer's disease in the animal models, and there are clinical trials of senolytics that address the SASP phenotype, and we are funding a clinical trial led by Miranda Orr at Wake Forest University of one of the first senolytic trials for Alzheimer's, combining a tyrosine kinase inhibitor called dasatinib along with an antioxidant called quercetin. And this is kind of the leading combination therapy from animal models on the senescent phenotype. And I think this is going to be the approach, because in the periphery, one of the challenges in this is in the periphery, you kind of want to get rid of these cells because they're inflammatory and they don't die, as you mentioned, they don't go into apoptosis. One of the theoretical things that have come up is that, do you want, neurons don't like to die either when they become senescent, but they might have still some connectivity, and one of the side effects could potentially be that dying neurons in the brain isn't the same as dying cells in the liver or the heart or the muscle, for example. But I think this is an important way forward in developing better biomarkers for the senescent cell phenotype. We have those in vitro and in vivo in animals, but we don't really have a good way of measuring them in the clinic. And I think that's right now, like in other targetable drug pathways, you know, we need to have biomarkers for senescent cell phenotypes in the brain that can not only identify people that have this phenotype, but also to monitor the effects of therapy in clinical trials. You know, mostly the way we're being able to do it now is through some exploratory biomarkers in the spinal fluid and inflammatory biomarkers, which might decrease with this combination therapy that's being used. So I think it's a really interesting way forward, but it's early days.
Irene, do you have any comments about the senescence interest these days too?
All right, I think the field is very interesting. It's advanced dramatically in the last few years. The piece I would add to that is what I'm surprised by is there isn't more in the field, and maybe the panel will note pieces that I don't know, is that there isn't more in the inhibiting the cell death pathways. There are very well described cell death pathways that we know are involved in models of all of the neurodegenerative diseases, and yet we haven't seemed to have made a lot of progress there. So maybe Howard would know more about what the current status is there.
Well, I don't know a lot about it, but I can tell you that caspase inhibitors were tried quite a bit, and I guess maybe that was side effects, but they didn't go forward really very far in the clinic. And that was one pathway that I know has been looked at for, you know, trying to, and there's not, and Rudy can tell me what he thinks, but I don't think there's a lot of evidence in pathology of apoptosis in Alzheimer's.
Well, more what I'm thinking of more is the stress pathways. So the ER stress and the J&K stress pathways is more of what I'm thinking of. And we just haven't, we know that those are have been shown to be involved in models of disease across the neurodegenerative spectrum, and yet not much that is advanced in the therapeutic space.
There is actually, and I won't mention the company, but I helped start a company that did a trial of one compound that would stop mitochondrial-induced apoptosis and protect the mitochondria. That's a taurine analog, TUDCA. And then the other compound was phenylbutyrate to hit the unfolded protein response in the ER. We combined them and we did an ALS trial, and it worked. And we published our paper in the New England Journal of Medicine in September, and then the survival paper just came out a couple of weeks ago, and now we're going for FDA approval.
Great.
ALS, and we're trying to get an Alzheimer's. It's in a phase 2 in Alzheimer's now. So that was like one of the first trials that actually did exactly what we were proposing, and we're keeping our fingers crossed.
Well, then those could have potential in Alzheimer's and PD and the other tauopathies and amyloidopathies as well. Then, right?
And Howard's helped funding the Alzheimer's trial, so thank you, Howard.
I see.
When I was a kid back in college, I had a guest lecturer, Hans Sellier, came to talk to us in my course. This was in the early 70s, guys, on the biology of aging. And I always viewed him as the father of aging. He's obviously the father of stress when it comes down to it from a biological perspective. And obviously aging involves a lot of stress too when it comes down to it. I always remember that fondly because it was an eye-opener in the early 70s, to be quite truthful.
Yeah.
One of the things that's different, I've been in the pharmaceutical industry for almost 40 years now, and entered about the time when the industry started looking at proteins as drugs. And I remember working for pharmaceutical companies who said, we don't make proteins as drugs. And needless to say, proteins are major drugs for the industry, and a lot of patients are feeling a lot better today because we are making proteins as drugs. But since that time, we have advanced our modalities even further beyond that. Now we've got nucleic acids, and we've got gene therapies, and we've got cell therapies, et cetera, too. Is there going to be a place for all of these newer modalities, which is also what we said about proteins many decades ago too, from small molecules, and what's going to be the most exciting thing in the next 10 years in terms of modality? Is it going to be small molecule? Howard, you said we got a problem with the need for too many antibodies in Alzheimer's. We have a limited ability to test some of the cell-based therapies. You know, maybe if we can generate small molecules to stimulate in vivo neurogenesis. Does anybody have a favorite in this case in terms of what's going to benefit patients in the future in terms of modality?
Yeah.
Well, I'd like to mention, you know, we all know that, if I may, I'm sorry to jump in, but, you know, we all know that ApoE4 is a huge risk factor, 20% of the population, 6% of the cases. And we've known that, I think it's almost 30 years now, and we haven't been able to drug it to figure out how to fix it. And I think that's because ApoE4 has so many biological effects that trying to affect one or the other, increase it, decrease it. The one thing we do know is that ApoE2 is neuroprotective, and people that are 2/4 have the neutral risk. So it really looks like ApoE2 can offset the risk of ApoE4. So what we've been funding over at Weill Cornell here in New York, and recently spun out a company called Lexio, is gene therapy for fixing ApoE4 by inserting the ApoE2 gene into an adeno-associated virus and injecting that intracisternally. And we're in phase 1 now in safety trials. It's been shown in non-human primates that injecting this adenovirus with the ApoE2 gene, that the non-human primates actually made ApoE2 in the brain. Of course, we don't know if this is going to be working, but I think it's going to be one of the first examples of trying to fix the ApoE4 problem in a way that kind of makes sense to me, because it's gene therapy for a genetic disorder in a way, which is basically the ApoE4. And so I'm pretty excited about that as a novel therapy. And I can also mention that ASOs are increasingly also being used, particularly when we see abnormal, like the C9orf mutation in FTD. There's some ASO strategies for that going on right now.
Barbara, one of the things that's attractive about gene therapy, apart from the fact that you can restore potential function in the setting of some type of deficiency, which almost always is a monogenic disorder, but it need not be the case universally, is that it's about focal delivery and being able to think about circuit dysfunction and the locus of those dysfunctions in neurodegenerative disease has made it possible to do gene therapy. I was involved in the initiative that led to a successful phase 1 study using AAV to express GDNF. A company was formed that's now being acquired, and it's progressing into later clinical development. And the thing that's very interesting is that despite the fact that most people would say, yes, neurosurgery is a very invasive process, it's very safe. We have looked over now more than a decade. When I chaired the Recombinant DNA Advisory Committee, one of the things I was most interested in knowing is how safe is it to deliver a virus vector that doesn't divide into the brain, and it's turned out to be very, very safe.
For ASOs, just make the point that, if you want to treat a symptomatic patient and turn down that microglial activation, neuroinflammation, we now have over a dozen Alzheimer's genes directly in the innate immune pathway. Howard mentioned TREM2, but CD33 and another one called NPP5D that makes a protein called SHIP1. These are genes that when they're turned up, they cause microglial cells to induce neuroinflammation. So we're trying to turn off CD33 with ASOs. We just published a paper with RNAi and AAVs. Or we're using antibody approaches to CD33. But I think we have to look more at these innate immune genes that are telling us what are the targets for hitting neuroinflammation, and hit them with ASOs or antibodies. And along those lines, I think it's just worth mentioning that we set up a screen for this in a 3D model, and just went through approved drugs, and we found that the old drug Chromaline, that's used for asthma, actually very effectively converted microglial cells from pro-inflammatory state back to their housekeeping role, clearing amyloid. So 2 for 1. So I worked with a company that did a phase 3 with a form of Chromaline that gets into the brain, a company called AZ Therapies, and our phase 3 trial just locked up, and we're going to have our data unblinded anytime soon now. So I'm keeping my fingers crossed and hoping that maybe we'll have the first drug that will convert the microglial cells away from neuroinflammation and get them to housekeep again and clear amyloid. And so stay tuned for that.
How we determine that endpoint, Rudy?
It's a phase 3 trial on cognition. So it is
it's a cognition readout.
It's some of the CD off some of the boxes, classic Alzheimer's year-and-a-half trial, 600 patients, micronized Chromaline that gets into the brain, and then the mouse studies, and then the dish studies. When it hits microglial cells, even if they're expressing CD33, So they're in this highly pro-inflammatory state, shooting out cytokines. It converts them right back to housekeeping phagocytic cells that clear amyloid. So we're hoping that translates into the human, but we'll know soon.
That's why it works so well in asthma too, originally when it came out
Well,
years ago.
well, it stabilizes mast cells through the Fc epsilon receptor.
Right.
We think in the microglial cells, it's working through the Fc gamma receptor, which is another way to convert the microglial cells out of the neuroinflammatory state.
Well, I think we would be remiss if we did not touch on a topic that none of you raised that I'm going to throw out here, and that is animal models of disease. We all sort of shake our head about them. So I'd like to know how you all feel about them in terms of their use in the development of drugs.
We don't have an Alzheimer's animal model. We have an animal model for amyloid.
Right.
And it was the mouse model that threw this field astray for 2 decades, because in the mice, when we put in the early onset Alzheimer genes and they made amyloid, but they didn't get tangles, that led to everyone saying amyloid doesn't cause tangles. And it turns out it's because they're mice. They don't have the right isoforms of tau. So you have to humanize the mouse and put the right isoforms of tau in, then you'll have an Alzheimer's model. And I think that's being done now.
I think also what I particularly like is the iPSC models. I know it's in a petri dish, but at least these are differentiated from the patients that actually have the disease, and you can look at it in a rather unblinded manner as opposed to focusing on a particular gene that you think causes the disease.
Well, we should probably start to wrap up now and end with the question that we were asked to address at the close of today's session, and that is, how may Neuroscience Pipeline look in 2040? As we move to that, I'd just like to make one comment. In 1990, the United States declared the decade of the 90s the decade of the brain, which I thought was really great, and this is good, where all kinds of wonderful things are going to happen. However, it was only an act of Congress which said that it was going to be the decade of the brain, and it was all just to increase our knowledge base around the decade of the brain. Every decade since then, I've been thinking it's the decade of the brain, and here we are 30 years later. Now we know a lot more than we did before, but the question, we're not as far advanced as we'd all like to be. So the question we're being asked to answer now is, 20 years from now, what is the pipeline for neuroscience, the drugs that's going to be available for patients, look like? And I'll start with you, Irene, and I know what you're going to say.
Maybe not. I watched it sort of peripherally in the field, the MS field, and I was started investigating there prior to there really being the type of drugs that are available now. And there was a perception at that point in time that we're just on the cusp of things changing. And as we can see now, there are a number of drugs that are actually quite effective for at least subsets of the multiple sclerosis patients. I have that same feeling with the neurodegenerative space. We've learned it is the case that that we're a long way from where we need to be, but in the 90s we did learn a lot. We learned a lot about the genetics. We've learned a lot more now at this point in time. The animal models aren't sufficient, but we've gained a lot of knowledge about what's going on in each of these diseases. And I really do believe that by 20 years from now, we are going to have drugs that do slow down disease in each of these major indications. I don't personally think it's going to be one drug that's going to be the end all be all. I think we're going to have to find that first drug that has some small effect and that add on to it to make sure that we have a fully effective drug that gives real changes for the patients.
And Howard Fedorf, what do you think 20 years from now?
I believe that most human diseases are really biological network diseases. So there will not be a single node that will itself be the panacea to drug, and then ultimately to either reduce the propensity to manifest or correct manifestation. So I think we're looking at network-based drug discovery. And I think that will ultimately mean that multiple drugs will be administered as long as we know how to modulate networks. I think cancer is the sort of poster child for this right now. And I believe that the nature of those networks are going to be different. They'll be distinct for different classes of patients. And it will not just be about their chronologic age. I think they will have fundamentally different pathophysiology that will be druggable. Whether that will mean that they're all small molecules, I think unlikely. I think there likely will be combination therapies that will include perhaps gene therapies, and maybe even cellular replacement therapies. But the big goal here, I think, is to stop thinking that a single target is going to be sufficient And while it is challenging and is not where the industry has been historically, I think what the pathogenesis is telling us about now, informed in part by genetics, but also by epigenetics, is that we have to move to thinking about biological networks. There's their dysfunction and how to drug the network in order to produce things that correlate with clinical benefit.
And we have to thank oncology for allowing us to do combination studies.
Absolutely.
We were loath in the pharmaceutical industry to do them for decades. So
Exactly.
the future is being opened up there. Howard, thoughts 20 years from now?
Yeah, I think what we're kind of describing in terms of extrapolating from cancer is a model of precision medicine, where we'll find multiple subtypes of Alzheimer's patients. We've talked about neuroinflammation. Some have it, some don't. I think epigenetics, metabolic disturbances in the brain, vascular changes. So I think we'll be subtyping them. I think that the revolutionary advances that we've had in biomarkers in just the last 5 or 10 years with neuroimaging and blood tests now is going to pave the way to precision medicine for Alzheimer's disease, the same way it paved the way for cancer, where we don't think of tumors anymore as organ specific. We think of them as cells that we phenotype. And so I think we're going to have tremendous advances in biomarkers that are going to enable a precision medicine approach, not only to distinguishing all these different subtypes and characterizing patients by their biological phenotype, but also doing clinical trials on drugs that address those phenotypes. So we'll have, as was said, combination therapies that address multiple pathways. And I think that that's going to be the wave of the future. We're going to have combination therapies with precision medicine. And I think we will have drugs. And the last thing I want to say is, our field is at least 30, 35 years behind cancer. It took us a long time to learn how to do really rigorous, efficient, reliable clinical trials. And I have to say that for example, the aducanumab study was one of the first studies where we knew who the patients were who were enrolled. We had a reliable biomarker that was the target of the drug. The drug worked in its pharmacology. It removed all the amyloid from the brain. And the last piece, whether it was highly effective or not, is up to the FDA now, I suppose, in some ways. But, the point is that here in 2020, we can finally say we know how to really do the clinical trials, and it's just going to get better from here. And I think that's really going to accelerate what happens by the time we get around to 2040.
Rudy, I started with you today. I'm going to end with you today, if you don't mind.
I agree with everything that the panelists have said in answer to this question. Even my longtime friend, Howard Phillip, even though we have our differences about amyloid, I think that in 2040, they're going to look back at us and just be astounded at the fact that we waited for the brain to degenerate to the point of dysfunction before we treated it, that this reactive approach to neurodegeneration will seem absurd by then. And we will be well into a program of early prediction based on genetics of when you need to start with early detection based on blood tests and, if necessary, imaging or digital technologies. And then actionable items. Then at this point, you start hitting those early pathologies. And it's I like to say amyloid's a match, tangles are brush fires, they spread. CTE is a match, tangles are brush fires. You don't get CTE till you're 50, you play football at 20. So these things take time. So if you want to hit these early pathologies like plaques and tangles, you have to do it a decade or more before symptoms. And I'm not surprised if you wipe out amyloid in somebody who's already suffering from dementia, where neuroinflammation is in full bloom, that you're trying to put out a forest fire by blowing out the match. So I think we have to come to that realization, and the FDA has to work with us that the future will be early prediction, early detection, early intervention with combinations that hit plaques and tangles and the neuroinflammatory steps based on what these genes are teaching us. But right now, and if we do have patients who are already symptomatic in 2040, and we do have to be reactive, I think the only hope is to put out that fire of neuroinflammation. It's too late to blow out the match or the brush fires of plaques and tangles.
Thank you all so much for this discussion we've had today. And I look forward to a brighter future for everyone in the neurospace in 2040 too. Thank you so much.